Hello, everyone, and welcome to another audio podcast in this series, the Excerpts Equipoise chapter. This is a Shientist, and this is another Storm Lobostics production. And I'm going to provide an excerpt around pathogenic mechanisms of cyst formation and kidney enlargement in autosomal dominant and autosomal recessive polycystic kidney diseases, and therapeutic approaches with EGFR tyrosine kinase inhibitors. The excerpt around this is thus. So the introduction: autosomal dominant polycystic kidney disease (ADPKD) remains one of the most frequently inherited diseases in humans. This disease is characterized with hyperproliferation of epithelial cells and results in the enlargement of fluid-filled cysts. Epidermal Growth Factor Receptor, or EGFR, remains associated with kidney cyst development and bacterial infections that can contribute to disease progression. The methodology around this work was the literature was reviewed in and around the research, and was identified using several databases, including PubMed Central, Medline Search Engine, and Google Scholar. Sort of the main sort of terminologies that were implemented to identify literature included polycystic kidney disease, epidermal growth factor receptor, tyrosine kinase inhibitor, renal cyst formation, and renal cyst infection. The studies beyond any studies or investigations beyond 10 years, over a decade, were not included in this research, in this work. The analysis around this was that this the cross-examination of research papers allowed to analyze literature around the role of epidermal growth factor receptor in ADPKD, in ADPKD pathophysiology, and wherein EGFR TKI as treatment of ADPKD, and the role of microbial cyst infections in disease progression. The discussion around this was that where ADPKD is concerned, there is a small body of literature. There is literature that has highlighted a sort of a disturbed link between EGFR tyrosine kinase inhibitor and cyst infection over the last decade. The crosstalk between SRC kinase and EGFR was observed, or has been observed, to have an impact on cyst development and progression of ADPKD. Combined treatment with different compounds inhibiting different pathways can be a successful approach in the treatment of ADPKD. There has been an observed relationship between cyst infections, decrease in kidney function, and PKD G mutations. Endotoxins of gram-negative bacteria are also involved in the development and progression. And conclusion around this work included the understanding of mechanisms of ADPKD, and several cancers have led to the identification of molecular targets as one of these. And one of these also surrounds epidermal growth factor receptor. However, studies reviewed and identified were highlighted small cohorts small cohorts and data highlighted there is a requirement or a need for broader and more collaborative efforts. And there is a need, also a need for further investigations or study to establish the role of endotoxin in ADPKD development and its influence on EGFR. The terminologies around this work the terminologies around this work include PKD, TKI, kidney care laboratory practice, EGFR, in context of epidermal growth factor receptor, microbiology, and science. The acknowledgements around this are to Orchakowska, and where there was a collaboration between an academic institution and the Renal Patient Support Group, the ARPKD. The excerpt, once again, was pathogenic mechanisms of cyst formation and kidney enlargement in autosomal dominant and autosomal recessive polycystic kidney diseases, and therapeutic approaches with EGFR tyrosine kinase inhibitors. Once again, this has been another Storm Robotics production.